Human papilloma. Hello everyone, our topic for today is human papillomavirus. Our biotechnology solution will be based on a cervical cancer focus. Human papillomaviruses are responsible for the most common STIs worldwide, affecting both men and women. They are non-enveloped, 8,000 base pairs long, DNA viruses, and encased in a 55 nanometer capsid. The structure of the genome can be divided into three major portions. A 4 kilobyte early E region that encodes non-structural proteins such as regulatory genes, a 3 kilobyte late L region that encodes two capsid proteins, and a 1 kilobyte non-coding long control region that contains a variety of cis elements which regulate viral replication and gene expression, or cutaneous infections, and are designated high or low risk depending on the propensity for malignant progression. HPVs can infect basal epithelial cells of the skin or the inner lining of tissues, with infection sites ranging from the mouth, throat, respiratory tract, or anogenital epithelium. The HPV is spread through contact, as the virus is spread through shedding skin cells and the virus enters through wounded skin. A big concern is the development of cervical cancer. Epidemiological studies suggest that the risk of contracting genital HPV infection and cervical cancer is influenced by sexual activity, particularly the link between genital HPV and cervical squamous cell carcinoma. Cervical cancer is the third most common cancer in women and the second most common cause of death from cancer in women worldwide. The papillomavirus infects the basal epithelial layer. The life cycle of HPV is directly linked to the differentiation of skin layers. The virus infects through cuts via the squamous and granular to the basal layer where the cells are differentiated and the virus is shed. The production of viral genomes is critically dependent on the host DNA synthesis machinery. In basal cells, HPV replication begins, and the viral DNA is maintained at a low copy number whilst remaining episomal in the nuclei of the infected cells. The cells undergo differentiation and move toward the epithelium where the virus replicates to a high copy number. Different HPVs may infect distinct target cells and consist of differences in the persistence of viral genomes in infected host cells. The E6 and E7 oncogenes bind to p53 with the help of p21, leading to retinoblastoma gene products to regulate cell growth as tumor suppressor genes. Together, these products deregulate the host cell growth cycle by binding and inactive tumor suppressor proteins, cell cyclins, and cyclin-dependent kinases. When HPV integrates into the genome, it enters common fragile sites and the host cell factors interact with long control region of the HPV genome, beginning transcription of E6 and E7. The E6 proteins are approximately 150 amino acids long and contain two domains related to the E7 carboxyl terminus, particularly the high-risk HPV E6 proteins that induce p53-like functions. The E7 proteins are approximately 100 amino acids and they modify the function of cellular protein complexes to elicit oncogenic activity. Studies indicate that continued E6 and E7 expression in cervical cancers are necessary for maintaining the malignant phenotype. The integration of high-risk HPV genomes represents the HPV E6, E7 induced genomic instability, putting it at an evolutionary disadvantage compared to the low-risk HPVs. Low-risk HPV, E6, and E7 proteins critically contribute to the viral life cycle, but they have sustained a sustainably lower transforming activity and do not induce genomic instability. Cell growth is regulated largely by two cellular proteins, the tumor suppressor protein p53 and the retinoblastoma gene product PRB. Unlike in many other cancers, the p53 in cervical cancer is usually wild-type and is not mutated. The HPV E6 gene product binds to p53 and targets it for rapid degradation via cellular ubiquitin ligase. Low-risk HPV E7 proteins bind to PRB at a decreased efficiency and do not induce PRB destabilization. The low risk of HPV E6 proteins do not efficiently interact with p53 and are incompetent for p53 degradation. However, they have an evolved life cycle to rapidly produce copious amounts of virus for host transmission.
HPV is a common DNA virus, which is mainly spread by genital contact. HPV infection can cause genital warts and several types of cancers, such as cervical, oral, penile, vaginal, vulva, and anal. HPV can be asymptomatic. That's why some people have HPV but never know. And that means they can continue spreading the virus through the population. But there are different types of strands of HPV. For example, HPV types 6 and 11 cause approximately 95% of genital warts. The warts can be flat or raised, single or multiple, and clustered. There are 14 types of high-risk genotypes of HPV cause 80% of cervical cancer. People who are infected by these types of HPV are usually asymptomatic, but some can experience pelvic pain intermittently or during sex, bleeding after sex, abnormal periods, and vaginal discharge. How to diagnose HPV? For female, they can do cervical screening to diagnose HPV or cervical cancer. Pap smear is a test to screen any abnormal cervical cells in the cervix. Pap smear method is to insert a speculum into the vagina to take samples of the cervical cells. WHO recommend women aged 18 to 69 years old who have ever been sexually active to do the test every 2 years. If cervical cancer is suspect, there are further tests for diagnosis. Diagnosis requires a colposcopy and a cone biopsy is performed to remove the precancerous or cancerous cells. Colposcopy identifies where abnormal cells are located in the cervix, and a cone biopsy removes a cone-shaped piece of tissue from the cervix, so you can obtain deeper layers for cervical cells for testing. According to Australian Institute of Health and Welfare, there was 933 new cases of cervical cancer diagnosed in 2020. The age standardized incidence rates will remain similar at 7 cases per 100,000 females. Back in 2016, there were 889 new cases of cervical cancer diagnosed. The statistics shows cervical cancer cases are slightly increasing. Compared to global age standard mortality rate, Australia is 1.7 per 100,000 females. Vaccines are available to prevent HPV infection. Gardasil is the most effective way to prevent HPV. Then, in 2014, Gardasil 9 vaccines released, and the vaccines can against infection for genital warts and cervical cancers caused HPV strands. So, how does Gardasil work? Gardasil contains structural and purified inactive virus-like proteins, VLP. These proteins can stimulate the immune system to attack HPV virus. The immune system will recognize viral proteins in Gardasil as foreign, then will develop antibodies to against viral proteins. The antibodies can provide immunity from future infections. Gardasil 9, introduced in 2018, protects against 9 strains of HPV that cause over 90% of cervical cancers in Australia. And HPV immunization is recommended for people approximately in year 12 to 13 at school for free. And people under 20 years old can get 2 doses of HPV vaccine for free under the National Immunization Program.
Despite this incredible preventative strategy, the prevalence of cervical cancer still attributes to many mortalities worldwide. But would you believe that some clever researchers in Queensland, as reported last year, cured mice with cervical cancer tumours containing HPV with an 100% survival rate? And that these scientists from Griffith University are working towards human trials within the next 5 years? Macmillan and his team were researching how to use CRISPR-Cas9 as a therapeutic gene strategy against cancer. This mechanism works by manufacturing a guide RNA, which is specific for binding with the host target DNA for editing. The guide RNA is a single RNA composed of CRISPR RNA, which is complementary to the target DNA, and the tracer RNA, which has a stem loop structure allowing binding to the Cas9 nuclease. The Cas will bind to a specific protospacer adjacent motif and cut 3 to 4 nucleotides upstream of this. Following cutting, there are 2 strategies for editing. One is non-homologous end joining, which is a very error-prone system which generates indels at random sites to disrupt the reading frame. The other is homology-directed repair, where researchers would supply a pre-designed sequence to insert for disrupting the DNA. So the CRISPR-Cas9 vesicle will enter the blood and then enter the target cell. The Cas will cut and modify the gene, and here we can see deletions. We know cancer is a product of unregulated oncogene and tumor suppressor gene activity, and the biggest challenge with designing a genetic cure is knowing which of these genes to target. So cervical cancer was chosen, as they knew tumorigenesis was driven by ongoing expression of HPV's E6 and E7 proteins, which modifies p53 and retinoblastoma activities. Challenges to be considered with CRISPR-Cas9 include ensuring safety and efficacy, and being able to reach target organs with minimal systemic toxicities. Also required is a stringent on-target gene specificity and no long-term adverse effects. The team compared numerous variables, including comparing wild-type Cas9, which is considered highly promiscuous due to ability to bind to off-target genes, and foci to Cas9, which is more stringent due to the Cas9 being catalytically dead and the foci nuclease domain requiring dimerization for the double-stranded breaks. They also compared the two methods of introducing a mutation. To test this, guide RNA expressing plasmids were designed to target two sites within the E6 or E7 of both strains. These were transfected into cultured cervical cancer cells, and along with either a foci to Cas9 or wild-type Cas9 expressing plasmid. Through assays assessing cell growth, wild-type was more efficient. This is supported by a Western blot showing wild-type to have greater restoration of p53 and retinoblastoma activities. Furthermore, the non-homologous end-joining editing activity was far more prevalent than homology-directed repair. Now it was time to test clearing cervical cancer tumors within mice, in which they targeted the E7 protein of both HPV 16 and 18 strains. The guide RNAs and wild-type Cas9 were packaged into pegylated lipoplexes, which just means attaching polyethylene glycol polymer chains to the lipoplex. This protects the nucleic acid content, as liposomes are usually destroyed by mononuclear phagocytes. The pegylation shields the liposomes by blocking the binding of plasma opsonins, and researchers determined stability in the blood for up to 6 hours, giving a safe delivery method capable of reaching the target organ. In HPV 16 positive mice with tumors of 20 millimeters cubed, 3 doses of 10 microgram of plasmid DNA cleared the tumors of 4 out of 5 mice and gave a very significant survival advantage. In HPV 18 positive mice with tumors of 1,000 millimeters cubed, the cancer was completely eliminated with 7 doses of treatment.